Hey, Jeff? Huh? Mute off. Jeff,
Yeah.
what is happening is somebody else is signing in and setting up a meeting and keeps kicking me off.
Same.
So,
Oh, it's I know it's Karen's trying to get logged in.
but she can't log in on the same account.
No, she
So that's what the problem is, and we need to get hold of her and let her know she has a different account or something.
yeah, yeah, I think she's probably just using the other login.
Yeah.
Let me see. I have her number somewhere.
Okay.
Yeah, it looks like she logged out.
Okay.
At least I don't see, she was on here, but she's not now.
Okay.
If you wanted to change your name, your screen name on there, you go to the participants and then touch your name, click your name, and it should give you some options to rename if you want to. I mean, you could leave it. You don't have to change it.
Yeah.
Sears. Kent wanted to be Katie and Banana for some reason.
No.
Hey, I fixed
But
it.
he wanted to be Banana, so I wrote Katie
Put
and
it
Banana.
in.
And then he kept trying
Anybody
to
else
change
getting
it to
kicked out?
Zoe and Banana.
Yeah, somebody else was signing into the account.
Gotcha.
Yeah, she just probably went in just like I did. I signed in before you started, and then it popped right in the meeting and said, "You're the leader of the meeting." I was like, "Oh, whoops! I didn't mean to do that." So I had to sign out and use my own personal account. I have to sign out of the SIM account,
Gotcha.
use my personal account, or else it doesn't work.
Yeah.
I think she may have got it figured out.
Yeah.
Point.
Hopefully.
You're out.
And you want to probably go find under meeting settings. If you can get to your meeting settings, you need to find the one that says chat with host only.
Well, where is
It's
it?
already on there.
Settings.
You can only do it to everyone or host.
No, she can control it. That's what I just do it to be to the host. I mean, I suppose if you want to keep everyone, so if somebody wants to, if you want to let the students ask questions that everybody can see via chat, that's fine too. Just it is already not allowing peer-to-peer chat, so
Okay. All right.
okay.
It allowed it on the computer last time.
Yeah, well, there were some messages up here when the craziness was going on, but that's. Yeah, but that's things going to everyone, so I guess that's all right. Okay. All right. That should fix it. I think we're missing a few. Hello, William. You just popped up there. And Katie, Asher and Olson, Jacob. All right. All right. I guess we've got everybody else in there.
We're just missing Elizabeth and Sarah
Yes.
and Danita.
Yeah. I don't know. She didn't sign in last week, so she may not be doing this. Okay. First of all, is everybody doing okay?
So far, so good.
Yep, doing fine.
We're
Pretty
good.
good.
How about you?
Yeah, we're doing fine.
Good.
So, all right. Let's go ahead and start with prayer. Father, we thank you for this opportunity to meet together and discuss your creation. We thank you for putting us together in this marvelous way that we can see the ingenuity of your design and how everything, even the tiniest processes in the cell, work together amazingly. We thank you that that you love us. We thank you for the cross, for sending your son to die in our place that we might come to know you as our Savior. We thank you for the resurrection and the glorious hope that we have in the Lord Jesus. We pray that you would be honored in what we do. We pray for our country at this time also, and for those who are ill, for those who are out of work and are struggling. And we pray that you would guide our leaders and in how to take care of the situation and guide us in how we can help others. We thank you again for your love. In Jesus' name, amen. Okay. So, to get started, let's see. There are some of you that are still missing work from Module 13. And Module 14 is looking pretty good. But I did get all of the grades entered that I have on the website. And the thing that I'm missing from some of you is your quiz grades, or how many you missed on the quizzes. And I need to know that so I can enter your grades for that. So check the website, see if there's anything that you guys are missing, and see if you can get them emailed into me. Okay. Somebody else has a big puppy dog. All right. So let's go ahead and first of all go over our quizzes.
We didn't get a quiz.
I don't think anybody got a quiz.
I didn't send it. I'm sorry.
No.
All right. Well, I'll send it. You guys can just take it and send it back to me and I'll grade it. I was busy sending a lot of things yesterday. Forgot the quiz.
Like you'll send it with our tests?
Yeah. Oh, that was another thing I was going to talk to you about. Instead of mailing out your test this time, I think I'm going to email it, just because I think it would be easier. So you can email them back, because the mail thing is not being real reliable. I know some of you guys are mailing things and I'm not getting it for several days. And others of you are not getting the test for several days. So I think it would just be better, we'll just email it. But please, after you print your test off, please delete the file so that we can maintain the integrity of the test and not end up with other issues.
Yeah.
Okay. All right, apparently the Harrisons are having computer issues. Well, that's good. We've all had computer issues today. So when you get the test, your mom or your dad needs to be the ones to be printing it off, okay? And you guys just kind of ignore it until it's time for you to take the test. Okay? So let's go ahead and we'll go on to our PowerPoint and go over our lesson for today. There it is. Kaboom. Share screen. And this screen. And let's go to the slideshow and play from the start. All right, so continuing on with the respiratory system. So last time we talked about the anatomy. By the way, you're not going to have any pictures to label on the test because really, what would you label? You could say lungs. That's pretty much it. Well, I guess trachea and voice box, but not very much to it. So only, there's no drawings for labeling on this test. So we talked about the functions of the respiratory system. Do any of you remember what those are?
Oh gosh, I'm trying to blink. I mean, obviously there's, you know, breathing, ventilation.
Yeah.
Its voice is another.
Yeah. Ventilation and voice.
The other two are respiration and olfaction.
Okay.
Get out.
All right, go back up there. All right, I'm gonna close that screen. Okay, yeah. All right, so voice, olfaction, ventilation, and respiration. Okay. And we talked about the stages or steps in respiration. First of all, there's external respiration that occurs between the alveoli and the blood, and this happens in the lungs. And then there's the gas transport that happens in the blood vessels all over the body, where oxygen and carbon dioxide are being transported around. And then there's the internal respiration that happens between the cells and the blood, and this happens in the tissues. Okay, so we're going to get into a little bit more detail in some of these respiration steps today. Okay, and it got a little bit technical in there in this portion of the module. We're talking about Dalton's law of partial pressure. And what is that actually measuring? The partial pressure.
It's the pressure or how much of a specific gas there is in a total volume of gas, if that makes sense. It's the best I've got.
Okay. Yeah. So it's basically the concentration of the gas in the mixture of gases. So when we're talking about the partial pressures, we're talking about a mixture of gases, and each one of the gases in there has a partial pressure. They're exerting a pressure depending on how much is in of how much of a particular gas is in the mixture. So the particular gases that we look at when we're talking about the respiratory system, of course, are oxygen and carbon dioxide. And these concentrations are going to vary at different points in the process of respiration. And all of this indicates which gas will be left in the bloodstream and which will be picked up. Well, maybe not left behind wherever it is, and which will be picked up. Okay, so you have a diagram like this. I think it's figure 14.8 in your book, where it talks about the partial pressures. The P stands for pressure, the O stands for oxygen, or O2 stands for oxygen, and P, pressure of carbon dioxide. So when we breathe in air, of course, hopefully you remember that the composition of air is about 78% nitrogen and about 21% of oxygen, and then 1% of the other gases. So the pressure of oxygen when we breathe air in is going to be 160 millimeters of mercury. The pressure of carbon dioxide, since there's just a small portion in the air, is only going to be like 3 millimeters of mercury. Okay, so that's when it comes in. Now, remember, we've got some residual air remaining in the lungs, and that's going to be more heavily concentrated with carbon dioxide, so that is going to reduce the partial pressure. So by the time that inspired air gets down into the alveoli, the pressure is going to be at, for oxygen, would be at 104 millimeters of mercury, and of carbon dioxide of 40 millimeters of mercury. So you see that there's a big jump in the amount of carbon dioxide present and a slight drop in the amount of oxygen present. But that's okay. It's that's still a very, very efficiently designed system. So then the exchange is going to happen there. Then when the blood is leaving the tissues and entering the lungs, the oxygen has been left behind in the cells and in the tissues. So the pressure of oxygen is going to drop down to 40 millimeters of mercury, and of carbon dioxide of 45 millimeters of mercury. So that's the blood that's going back to the heart. Back to the lungs to be reoxygenated. So that's deoxygenated blood. When blood is leaving the lungs and entering the tissues in the tissue capillaries, the pressure of oxygen is going to be at 100 millimeters of mercury, and the pressure of carbon dioxide at 40. So remember diffusion. So because of these differing levels, when the deoxygenated blood gets to the alveoli, there's going to be a lot of carbon dioxide in the blood, 45 millimeters of mercury, and in the alveoli, it's not going to be that much. So the carbon dioxide is going to be coming out of the bloodstream, diffusing out to get into the alveoli, and the oxygen is going to be coming out of the alveoli to get into the capillary so that it evens out the pressures of the oxygen. Okay, and so that is why oxygen is absorbed into the bloodstream in the alveoli and carbon dioxide is left behind. It's also why when you get down to the tissues, and there's these tissues that are very needy of the oxygen is going to leave the bloodstream, go to the tissues, the carbon dioxide is going to leave the tissues and go to the bloodstream. It's all because of diffusion. Okay, is everybody clear on that? There are not going to be a lot of technical tests. There's not going to be any technical questions about the partial, this much partial pressure is here and that much partial pressure is there. That's just mostly so that you can see what's happening and why, and you understand it. Okay, the control, okay, now let's go change topics. We're going to respiratory control. And what causes us to be able to breathe like we do. When we're sleeping, we're breathing and we're not even thinking about it. Sometimes we think about it when we're awake. And we have some voluntary control over our respiration, because if we think, I'm going to pant like my puppy dog, we can do that. Or if we think I'm going to take a nice deep breath here, we can do that. So we have a certain amount of control over our breathing, but we can't decide we're going to hold our breath forever, because that's going to be overridden by our other systems in our body. So the thing that controls our ventilation is some of these little neurons that are in the medullary respiratory center of the medulla. There's a couple of groups there. There's the dorsal respiratory group, that's groups of neurons in the medulla. And the dorsal respiratory group is going to be involved in controlling the diaphragm, telling the diaphragm when to contract and when to relax. During exercise, especially, the dorsal group is going to send those action potentials to the abdominal and intercostal muscles as well, so that you can breathe deeper. There is one reflex, the Herring-Brewer reflex, that is actually going to prevent the overstretching of the lungs. So if you're taking really, really deep breaths, there's going to be, there is a monitor in the lungs that is going to say, okay, that's all, we can't go any further, and it will send action potentials to cause you to relax the diaphragm and breathe out. Okay, so it's not going to allow your lungs to be overstretched and popped. So we can't pop our lungs like a balloon. Okay, then there's a ventral respiratory group, and these neurons set up the basic rhythm of breathing. So your breathing usually follows a certain rhythm, certain number of times per minute that you breathe. If you're sitting, it's going to be a slower rhythm than if you're running, but that basic rhythm is set up by the ventral respiratory group. And then there's the pontine respiratory group, which is located in the pons. And these are neurons in the pons, not pons, okay? And these are going to aid the switch between inspiration and expiration, okay? Because you're breathing in, then you have to breathe out. The thing that switches from one to the other is going to be those neurons in the pons. So this portion of the brain is the one that's going to be responsible for this very vital ventilation. Okay. There are also some chemical control systems that affect our ventilation. In fact, the carbon dioxide levels and their effect on the pH are the major controller of ventilation. Okay, that's in bold print, so it means you probably need to understand that. So the thing that's going to have the major control over ventilation is the carbon dioxide levels. And the carbon dioxide levels are going to affect the pH of the blood. And how does that happen? First, whenever we have carbon dioxide and we dissolve it in water, it's going to cause the formation of what we call carbonic acid, H2CO3. And you see how that can happen. You see the hydrogen that has bonded to the carbon dioxide, and then there's this extra oxygen molecule. So we have carbonic acid, and of course an acid is going to raise or lower the pH?
Lower.
Lower, because acids, remember, have a pH of below 7. So then the acid is going to release a hydrogen ion, and it's going to form a bicarbonate ion. So acid dissociation has the effect to lower the pH because a bicarbonate is a base. So we go from carbonic acid to the bicarbonate to lower the pH. So these kinds of chemical reactions are going on with the carbon dioxide in our blood, affecting the blood pH. So the more carbon dioxide you have in the blood, the lower the pH of the blood. So carbon dioxide is removed from the blood by increased depth and rate of ventilation. So if your blood pH is going a little bit too low, then you're going to be increasing your depth and rate of ventilation to get more carbon dioxide out of your blood. Now, if you have a high pH in the blood, if it's getting over toward the basic level, then there's going to be decreased ventilation, so you can keep more carbon dioxide in the blood because that forms the carbonic acid that brings the pH back down. Now note, the respiratory system is the second major control system for regulation of the pH of the blood. And you remember that the pH of the blood is a very narrow range, 7.35 to 7.45, very, very narrow range. So it can get out of whack very easily. And so we have the respiratory system as a system to regulate that, but it's the second one. The next next module, we'll talk about the major control system, the number one control system for the regulation of pH of the blood. And that's going to be the urinary system. And the reason that is important because the urinary system is controlling the chemical levels of a lot of different chemicals in our bodies. So we'll find out more about that starting next week. Okay. Then there's some other chemical or chemoreceptors in the medulla, the medullary chemoreceptors that are going to be also involved in this process. So these are going to be affecting the rate and the depth of the ventilation. So when the blood pH goes down, CSF, what is CSF?
Cerebrospinal fluid.
Right. So when the blood pH goes down, the CSF also goes down. And the chemoreceptors, excuse me, are going to be sampling the CSF and doing like a test on it to see what the pH is. If the pH is low, if the pH is low, then it's going to increase the rate of ventilation. If the pH is high, it's going to, I've got a thing on my screen, don't I? Right. If the pH level is high, then it's going to decrease the rate of ventilation. Okay. So it's the chemoreceptors that are testing the CSF that are going to determine if you need to breathe more or breathe less. There are also chemoreceptors that sense oxygen levels. Can you bring me some water? And these are located in the carotid and aortic arches. And they're there and they detect the oxygen levels. And if the oxygen levels are a little off, they will increase rate of ventilation as well. These, though, are seldom stimulated because the efficiency of the pH system. That system works so well that you don't really have to involve these other chemoreceptors, but they're there just as another mechanism to keep our bodies functioning and safe. Okay, now is the time to talk about cellular respiration. Now that we've got that oxygen in our system, what is our body going to do with all of this oxygen? Thank you. So you might want to get out your little charts that I sent to you. You did get that one, didn't you?
Yes.
Cellular respiration. Okay. And we're going to go kind of over this nice and slowly. Where did it go? Oh, here's my chart. Okay. So that we're sure that we've got everything, or that we're understanding all of this. Okay, so we've got this nice formula here. Who's making lines on my page? Okay. this formula. And notice one thing up here called specific enzymes. And this is one thing we're not really going to study in depth in this class. If you take biology or an anatomy and physiology class in college, you will get to know these things. We're not going to learn the names of all these enzymes, but you need to understand that at every step of the way, we have enzymes that are working to make this equation happen. It would not happen if we did not have these. So in order for this reaction to occur, we have to have all of the reactants and we have to have the enzymes. Okay, so it's not just going to happen without the enzymes there. Not going to just happen on its own. All right, so the reactants are glucose and oxygen. Now we're going to start with a glucose molecule. The oxygen molecules get added in later on in the cycle, but they're not going to be involved in the first stage. So once we complete this reaction, we're going to end up with 6 molecules of carbon dioxide and 6 molecules of water and 36 ATPs of energy. So there's 4 stages. All of them are enzyme controlled. Now this is cellular respiration. Don't get it mixed up with respiration, general respiration, I guess we could call it. So cellular respiration has 4 stages. And these are glycolysis, oxidation of pyruvate, the Krebs or citric acid cycle, and the electron transport chain. Okay, so first one let's look at glycolysis. Now glycolysis occurs in the cytoplasm. And in order to get this reaction started, we're going to need some ATP. Now, notice down here, here it says glucose. This is what it is. And glucose is 6 carbons, 6 oxygens, and 12 hydrogens. Okay, we're just going to follow the carbons through this just to make it easier to keep up with. Okay. So we're going to have 2 adenine triphosphate. Here are 2 little energy packets. And they're going to be broken apart. And one of these phosphates, and this is what the PI stands for, is a phosphate. One of these phosphates is going to be stuck on each end of the glucose molecule. Okay. Here's, by the way, I have our glucose molecule here. You can't see it too well, but it's got the 6 oxygens, 6 carbons, and 12 hydrogens. Okay, and I'm going to set it over here to the side of my table because that will help us keep up with some things as well. All right, so the first step of this glycolysis reaction then is going to give us, we still have our 6 carbons here. So this is called fructose diphosphate. There's a phosphate here and a phosphate here. And remember, fructose and glucose have the same formula. They're both C6H12O6. They have a little bit different structure into the molecule. So the molecule structure has changed a little bit here, but we're ending up with this with the 2 phosphates on the end. And we're also ending up with 2 molecules of ADP left, which is what we have left when we use ATP. ATP is Oh, not TP like toilet paper, but toilet paper's on our mind a lot these days, so that's why every time you see that. Never mind. All right. So when you see ATP, that means there's 3 phosphates. ADP means there's 2 phosphates in the molecule. So we've taken one off of each of the phosphates, and that's left us with 2 ADP. All right, now we're going to bring this thing that we made in this reaction, and we're going to bring it down here to step 2. And you'll see that throughout this whole diagram that we have of cellular respiration. We're going to take what we made here, bring it to the next step. All right, so we've got fructose diphosphate, and what we're going to do with this is we're going to just break it in half. And so we've got the 3 carbons here. Phosphorus are still there because we've got this little subscript there too. So we've got 2 of these molecules made up of 3 carbons and a phosphate group. That's called PGAL, or phosphoglyceraldehyde, I believe is the long name for it. Okay, so this is where we're at. We've got PGAL. Let's go to our next stage. So 2 more, here we go. We brought it to step 3. We made it in step 2. We're bringing it to step 3. All right, now we're going to add 2 more phosphorus molecules. And then we're going to add this new little ionic compound here called NAD. It's I'll have to look at it just a second. Oh yeah, nicotinamide adenine dinucleotide. And that's who this guy is. And this guy is a hydrogen acceptor. It really, really does like hydrogen molecules, and it will do anything to get hydrogen molecules. And that is what the big deal is in going through all of this, is that we're going to end up with hydrogen molecules. So we have the NADs here, and they're going to enter into our reaction. So we've got, oops, sorry, went too far. So we've got our PGAL here, 2 phosphorus and 2 NADs. That's our reactants. And they're going to end up taking away some of the hydrogens. And so we're going to end up with 2 diphosphoglyceraldehyde. Okay, so 2-DPGA. We've lost the L there because our hydrogens are leaving. So this new compound is going to look like this. It's got 2 phosphorus, 3 carbon molecules, and then we've got the NAD molecules that have taken away some of the hydrogen molecules from our original molecule over here. So we had 12 hydrogens. Now 2 of those have been taken away. So 2 of those have been taken away by these NAD molecules, and those are going to be set aside until later in our reactions. Okay, and so now we've got this compound here, the 2-DPGAs. This is going to come down here. And we're going to add some ADP to that. So we have 2 of the phosphorus here. This is what we have here, which is the same as up here, and 4 ADP. So we actually have 4 phosphorus molecules available. Do you see that? And this is going to make 2 pyruvate molecules and 4 ATPs. So these ADPs pick up those phosphorus molecules, and that becomes ATP. So we're going to end up making some energy in this reaction. So we use 2 to get started. We're going to make 4, so there's a net gain of 2 ATPs of energy in this reaction. Now, all of this is happening in the cytoplasm. So you may wonder, where did these phosphorus molecules come from? Where did these NAD molecules come from? Those are molecules that are available in the cytoplasm of the cell, so that there's a rich store of materials for the cell to use in going through these reactions, because it does go through these reactions pretty continually. Now notice we haven't used any oxygen. This is an anaerobic, this is what we call anaerobic respiration, because we are able to produce some ATP through that. Okay? So any questions about glycolysis? All right, let's go to the next stage. Now, what do we have? We have pyruvate here. And so pyruvate is going to go into the next stage, which is called the oxidation of pyruvate. So we went through all of these crazy steps to get to pyruvate. And so now we're going to the next stage, which is going to occur in the outer membrane of the mitochondrion. Oxidation of pyruvate, it can also be called the acetyl coenzyme A formation stage. So remember the mitochondrion? We've talked about it before. It's the energy or power plant of the cell. So let's kind of use our imaginations a little bit and think about these pyruvate molecules. So here's the storyline. We were minding our own business, just going through the front door of the energy plant, when we were jumped by 2 NAD molecules who ripped off 1 hydrogen from each of us, causing our carbon chains to break apart. We totally lost one of our carbons. It turned into a vapor right in front of our eyes. Fortunately, 2 kind CoA molecules grabbed onto us and carried us to the matrix. Okay, so that's kind of what goes on in this. So we have these pyruvate molecules just kind of coming towards the door of the energy power plant there. And then here comes these 2 NAD molecules. And of course, remember, they love hydrogen. And so the first thing that's going to happen, okay, and they're being shepherded along actually by these coenzyme A molecules. The first thing that's going to happen is, oops, they're going to have a couple of their hydrogens broken off that are going to join to the NAD molecule and become NADH. So we've lost 2 more hydrogens. And that is going to cause the carbon chain to break apart. One of the carbons is going to be, or 2 of the carbons, well, 1 from each molecule, are going to be broken off and combined with oxygen. Now, where did this oxygen come from? Okay, remember this molecule, the glucose molecule, there's got 6 molecules of oxygen on it. So these 6 molecules of oxygen are available for use. And so some of them get used here actually 4 of them will be used here to produce carbon dioxide. And of course, that is a vapor that is given off. And so we're losing the carbons. We've got 4 oxygens that have been taken away from our original glucose molecule. All right, so this is the only thing that's going to happen in this. This little molecule here is called an acetyl molecule. It's going to combine with this enzyme, CoA, and it's going to form acetyl coenzyme A. Okay, so we're going to take that now. Here's our acetyl coenzyme A. And this is going to be the beginning of the Krebs of the Krebs cycle, or the citric acid cycle. Now, this acetyl coenzyme A goes into the matrix of the mitochondria, which is the middle portion. And here is step 1 of this process. So we've got the acetyl CoA, and then the 2, we're going to add to that some oxaloacetic acid, which is something that is going to be made or used and then remade. So we've got 2 carbons here, 4 carbons here, and we've got 2 each of these molecules. So we're going to end up with 2 each of these molecules of citric acid back to a 6 carbon molecule. And when citric acid is formed, then our coenzyme A is going to be released. And so we're not going to have that molecule anymore. It's going to be released to go back and find some more, or to combine with some more acetyl or help some more pyruvate molecules. Okay, now, so we've got citric acid here. We're going to bring it down to this one. This is a really long reaction. We're going to be adding in some ADP. We're going to be adding in some phosphorus. And here is another molecule. We've got some NADs coming back. We've got some FADs. FAD is also a hydrogen acceptor. Its name is flavin adenine dinucleotide. By the way, both of these are made up of B vitamins. So if you don't have a sufficient supply of B vitamins, you're not going to have a sufficient supply of these molecules. And so that's going to affect your ability to do cellular respiration and produce energy. So that's why those vitamins are some very, very important ones for you to be sure that you get. All right. Now, this is going to give us, wait a minute, where did that come from? I think I must have a wrong number here. Yeah. Well, I don't know how I've gotten 6 there when I only had.
On your chart, you have, oh, I just pulled it up.
Yeah.
You have 6 NAD in the first half of that instead of 2.
Oh, yeah. Okay. So I've got it wrong up here. All right. That's where it is. Okay. I'll have to go back and fix that. So that should be 6 right here. So ignore that too. And so we're going to end up with 6 of the NADH, and we're going to end up with 2 of the FADH2. And we're going to give make 2 ATPs of energy. So that comes about here. And then we're going to end up with 4 molecules of carbon dioxide. So we're going to use the last 2 molecules of oxygen from here, and then we're going to start on, or we'll use, let's see, 4 molecules of oxygen, no, 6 molecules, wait a minute, I can't, 3 molecules of oxygen. No, I'm confusing myself. 602, never mind, 12 molecules. All right, so we use 4 molecules of oxygen from our original formula where we had the 602. So we've got 4 molecules of carbon dioxide that are going to be given off. So we started with 6 molecules of carbon here. We're getting rid of, 12. Here we're getting rid of 4. Here's the other 8 molecules, reforming oxaloacetic acid, which will go back and be used up here. So we use it here.
No, it won't let me use that email because it's the same
Okay.
what Jean's using right now. So I have to have a different email. You said an account, right?
Yeah, Karen.
Oh, sorry. Am I?
Yeah, you're in our lesson.
Sorry. Yes, sorry. I was trying to get mine. Let me deactivate. How come I'm here? I'm
Okay. This has been an interesting day. Okay, so here's our oxaloacetic acid. Here it is down here. We're going to cycle it back up here. That's why it's called the citric acid cycle or the Krebs cycle, is because it's used here, remade, come and be brought back to be used here. All right. So now with that, what have we got left? All of the carbons are gone. All of the hydrogens have been taken up. All of the oxygens have been used up except for 3 molecules of O2. So all of the oxygens have been, all of the others have been left. So all we have are these things here, the NADH and the FADH molecules. Now, throughout this process, there's been these NADH and FAD molecules that have been collecting our hydrogens for us. And we're going to see them coming back. So, all of these molecules we have a total of 8 NADH molecules and 2 FADH molecules. And they're going to bring those hydrogens into the inner membrane of the mitochondria. And they're going to let them go. So all of these reactions that we've been going through have been for the purpose of getting these hydrogen molecules isolated so that they could be brought to the inner chambers of the mitochondria. So the electrons then are going to be traveling through a chain of 7 carrier molecules, and energy is going to be released with each change. And so the energy is going to be used to actively transport the hydrogen ions across this membrane, inner membrane. And the hydrogen ions are going to gather in the inner membrane space of the mitochondria, then forced by diffusion to go back through the inner membrane and into they're going to be going back and forth through this membrane. This is going to require special channels. It's going to have ADP on one side, phosphate molecules sitting on the other side. And as the ADP and phosphate are going to use the energy from the hydrogen ions traveling back into the matrix to combine, forming ATP. So the hydrogen ions have are releasing energy to enable the ADP and the phosphate to combine, forming ATP. And it forms quite a lot of it. This process brings about 32 molecules of ATP. So here's what it's going to look like. We've got energy from the hydrogen diffusion. This is step 2. And we've got ADP and phosphate to form ATP. 32 ATP produced. And then we've got to do something with this, the rest of this, with the hydrogen. Once it's done its job, the hydrogen is going to be combined with the remaining oxygen molecules to form 6 molecules of H2O. Okay. So let's summarize. In glycolysis, glucose is used up, 2 ATPs are made. That gives us the pyruvate. And so in the oxidation of pyruvate, we're going to be making 2 carbon dioxide molecules. In the citric acid cycle, 4 more carbon dioxide molecules are made and 2 ATP. Then electron transport chain, the oxygen is going to be used up, 6 water molecules made, and 32 ATP made. So the overall products from the reaction are 6 CO2, 6 H2O, and 36 ATP. Okay, starting with the glucose. Now I know this seems complicated, and sometimes if you study it real carefully, it seems like some of the numbers don't add up. But that's because there are other things that are going on that we're not learning about, which we should probably be thankful for. So if you in college classes, you will get into this into much even greater detail. It seems like there's a lot of detail, but this is still a rather simplified explanation. So if you think about it, so we've got this very complex reaction that has to have all of these different molecules available. That's certainly looks like something that was designed. That doesn't look like something that could happen from random processes. And this is essential for life, because if you don't have the ability to produce energy, the cells cannot carry out their functions. If the cells can't carry out their functions, then the cells cannot live. So this is a very essential process for life. And it is a very, very amazing process. And you are going to need to know this pretty well for your test. Okay, so you're going to have a paragraph that's describing this reaction, and you're going to need to be able to fill in the blanks of what happens where. On your test, there will be an answer box that goes with this portion of the test that's on a separate page. But it doesn't have all of the answers. Some of them you're going to have to know, like especially the names of the stages and especially where they occur. That would be something for sure that you would need to know, and that will not be in the Word box. Let's see. Okay, does anybody have any questions about any of that?
I don't think so.
Okay. The important thing is to see the products that are made and how they are used in the subsequent reaction over and over again. That's the way it works. You make something and then you make it to be used in the next reaction. Okay. So let's just do.
Asher water.
You know, he can hear you.
Okay. What is respiratory distress syndrome? Do you remember that?
Respiratory what?
Respiratory distress syndrome.
I remember reading about it, but I don't remember exactly what it was.
Who might get that?
Infant,
Okay.
if I remember correctly.
What kind of infant?
Premature. A very newborn.
Okay,
Yeah.
a premature infant. Okay, a baby that's born before it's fully developed, because the lungs are the last thing that gets developed. And they're missing what? Why do they have the syndrome? What are they missing?
Surfactant was
Surfactant.
one.
Right. Okay.
There's something else, but I forgot.
What is principal inspiration?
That's isn't that inspiration that you don't control?
Well, it's just it's just normal breathing. Okay. What is emphysema? I don't think we talked a lot about emphysema last week. Okay, a lot of times people that have been smokers for all of their lives, when they get to be older, they have emphysema. It happens because of the damage done to the alveoli of the lungs from the smoke inhalation, and that causes the alveoli to kind of clump together in large clumps. And because they clump together, they tend to not be as elastic, so you lose the elasticity of the lungs, and you're not able to breathe out as easily. So it's easy, you can get your breath in, but sometimes it's hard to get the breath out. So it causes a great deal of difficulty breathing. And also, if you've got the alveoli clumped together, you're reducing the surface area of the respiratory membrane of the lungs, which is where all of the transfer of the oxygen and carbon dioxide goes on. So that's a couple of issues with that. Now, that being said, there are other things that can cause emphysema. So you need to be careful about anything that you're breathing, like e-cigarettes. There's the new thing. Of course, those are making a lot of people sick now because of what they're putting in their lungs. But
Getting pneumonia.
But anytime you're breathing any kind of smoke type stuff into your lungs, it can cause damage to the alveoli, because those things are very, very delicate. So that's something you need to be cautious about. Being careful what you breathe in. Okay. What is forced inspiration?
When you breathe in and you suck more air into your lungs.
You breathe in forcefully.
Yeah. So it's something that you do willingly. You're forcing yourself to take a deep breath. So it's just a deep breath. Okay. So what is forced expiration?
When you blow as much air out as you can.
Okay. All right. What is pneumonia?
It's when your alveoli get filled with fluid.
Right. And if they're
Because of an infection.
yeah. If they're filled with fluid, then they can't work. So and that's what happens with pneumonia, is the alveoli are unable to function. So sometimes they'll put people on a ventilator, they'll give them oxygen to help them get over that. Okay. Let's see. Oh, here's something else we haven't talked about. And that is aspiration, or not like a goal that you set for yourself, but aspirating something. When you take something in by means of suction, like, for example, if you're maybe breathing while you're getting a drink of water and you breathe some of that water down into your airways, that would be aspiration. And in some circumstances, that can be a problem.
In the Ben Carson.
If especially with,
one of the biotrans.
they used to have these masks that you would put over your face when you needed oxygen. And if something got in that mask and you were taking that in, you'd be aspirating that stuff into your lungs. So that's what that refers to. All right. What is the functional residual capacity?
That's how much air your lungs can hold. No, sorry, that's total lung capacity.
Right.
Functional is
How much is left after you breathe normally?
Right. After a normal exhalation, that's the functional residual capacity. Because you don't breathe all your air out of your lungs. Even in a forced expiration, you can't get it all out, which is a good thing. Because we need our lungs to stay partially inflated so that they're easier to inflate the next time. I don't know, I still have that up there.
What's that button?
And show. Okay. 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 Um, all right. All right, so what is ventilation?
Breathing.
Okay. It just means we're getting process of getting air in and out of our lungs. The tidal volume of air.
The maximum amount of air that your lungs can hold.
Not quite.
It's the volume of air inhaled or exhaled while breathing.
Yeah. Compliance.
That's how easy it is, or how elastic your lungs are.
Okay. It's or how easy it is for them to inflate. Elasticity refers to the deflating more than the inflating. All right. Remember the pneumothorax? We did talk about that one.
Air in the pleural cavity.
Which leads to a collapsed lung.
Yeah, and it causes the lungs to collapse because the seal in the cavity has been broken. All right. You need to review those factors that aid ventilation and factors that aid, and some of them aid inspiration, some of them expiration. There's another one. Let me see. Here's my other question. Okay, also, why is it that our system of getting oxygen into our blood works so well? It's very, very efficient system. So even though a person might not be in perfect health, their blood, when it comes out of the lungs, is almost always 100% oxygenated. Sounds like a train just came in here. Why is that?
Because it has to be.
Well, yeah, but
I don't know. I don't know why.
What,
But if it wasn't, that'd be bad.
yeah, what makes it way? What are some factors in the way in the design of the respiratory system that makes it work that way? Okay, think of the air where the alveoli and the blood where they exchange. We have this very large respiratory membrane, remember? And so there's a very large area where the oxygen and carbon dioxide can be exchanged. And then the capillary walls are very, very thin. And that allows for oxygen to be diffused into the bloodstream easily. The shape of the red blood vessel, not blood vessel, blood cell, red blood cell. The narrowness of the capillaries that forces the red blood cells to go past one at a time. There's 6 of them. Well,
Is the alveolar fluid one?
I don't think surfact. No, I don't think that's necessarily one. Let's see. Oh, here it is. I got it. Oh, functional residual capacity. There's always some air available. And the other one is the controlled relationship between ventilation and blood flow. So that if you need more oxygen, you're going to breathe more, and it's going to speed up the process of the blood flow. So if you need more oxygen, you're going to breathe more, your heart rate's going to increase, and that's going to keep enough oxygen going into the lungs so that it can be taken care of. All right. So those are some things to review and study for the test. Study that chart. I will send you a study guide, and then I'll send you your test later today. So I hope all of you have a nice Easter. I know it'll be a quiet one anyway. So I hope you have a good Easter, and that it, even though it's kind of a quiet time, that it will be really meaningful to all of you, that you will just take in all that happened and all that this is about, and that it will have a good impact on your life. So you guys stay healthy, wash your hands a lot, and I guess we'll see you next week.
Yep.
Thank you. Bye.
Except for I'll see Olson on day.
Yeah,
See you on Wednesday.
we'll see you virtually next week, not in reality.
Yeah.
Okay.
Yeah.
All right. Well, God bless. We'll see you next time.
Thank you.
Bye, everybody. Happy Easter.
Happy Easter.